And like the title is said, Introductions to Genetics. Introductions to Genetics exam. The exam is on 16th. And I don't know shit. So yeah. So these are the only things I have as part of the. Okay. Describe three main foundations leading to discovery of DNA structure. What is the structure of chromatin? What are Ozaki fragments? Oh, Japanese desu. Ho ho ho, Ozaki desu ne. So ne. Name the most important types of RNA and describe them. Sketch how translation proceeds at ribosome and label main important components. What is a chaperone? Name at least one. Describe three main foundations leading to the discovery of DNA structure. What is the structure of chromatin? Ozaki fragments. Dude, it's repeating. Okay, these are the answers. Okay, describe three main. On. Describe three X-ray diffraction by Wilkinson fragments. Chargaff's rule, molecular dimensions, and bond lengths by Linus Pauling. Okay. And people, YouTube, I got an announcement to make. I think I told this more than 3 times. I bought a road bike. And today, oh shit. Actually, I should be making. Um, I should be making reels on important subjects for like 60-minute 60-second videos, short videos on explaining different terms, but I still haven't started it. I need to start that, but before that, today I will be recording myself heading to my place of work. It's gonna be good. Trust me, people, it's gonna be good. At least I will try doing it. What is the structure of chromatin? Example questions. I mean, what the. What are Ozaki fragments? DNA replication basics. Complementary, semi-conservative, helicase, primase, DNA polymerase, exonuclease, ligase. What are Ozaki fragments? Example questions. How many PDFs are there? How many pages? 19. It's doable. Important dates. I missed this one. Okay. What is chaperone? Name at least one example. Levels of protein structure. Mm, mhm, mhm, Okay, where's the big boys? We need the big boys up here. Big boys or big woman? No, it don't matter. Deep sick. Where's the big people? So GPT, I have an exam on introductions to genetics in 4 days. No, how is the... Mm, let me see the mega file, because maybe it's in the mega file. Today, how's the rain? No rain? Nope, there is rain. I just hope the rain is not on the time when I head to work. Because today, I'm going on a bike. Hell yeah, boy! It's a bike. You know, it's gonna be amazing. I'm done with the fucking trams. I'm done with people. No, I'm not done with people, but I'm just done with trams. I don't want to be a burden to other people. If I have the option to go through bike, I should use it, right? I should not accommodate the space in a tram if I have the option to not use the tram. And my the option that I'm using is not gas powered type shit. It's a bike, so it's a human powered mode of transportation. So there's no carbon emissions, shit like that. So I'm such a nice guy, you know. I'm such a nice person. Uh, yeah. Whoa, 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 whoa. Whoa, 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 whoa. Mm, mega file. Yeah, the group. Genetics PDF. There's a PDF for genetics. DNA versus RNA. Watson's okay. That's some good shit. Send to my laptop. There you go. Schofberger's exercise. During solvolysis, synthesized alkyl bromide products, substitutional elimination. I think this goes to the organic chemistry. Oh, 40. Dude, it's 46 pages. Okay. Yeah, there we go. No, not here. Here. Stop. I did have the PDF. Okay, I do have the Schulzberger exercises. It's been some time since I started writing on my tablet. Welcome back. Welcome back, tablet. It's been a while, innit? It's been a fucking while, eh? Lot of stuff to do, and let's do it. Inglés, yes. Por favor. So exam is on 29 9. Submission of the protocols. Oh shit. Oh shit. It's this month. I have to submit the protocols this month. Digital version is on 25th. Could do that. Okay, it starts. Physics 2 for biological chemistry. That's on 25th. I gotta take that. Organic chemistry 14.8. I'll take that. Introductions to genetics. I'll take that. Math. I want to take it, but it's not giving me the option. Or oh yeah. Where's math for biochemistry 2? Nope, nope. No. JKU. Get a bit shorter. Purine nucleotides, pyrimidine nucleotides, X-ray diffraction data by Dawkins. Let me ask this question. Let's see what are these two guys' answers. Griffith transformation experiment. Is that mentioned here? Nope. Now, what is this guy saying? X-ray diffraction by Rosalind Franklin and Maurice Wilkins. That's correct. Chargaff's rule. Okay, you got that one. And Pauling's alpha helix model building. I gotta say, Deep Sea got this one. So let's start with you're referring to the more direct scientific foundation of DNS query. Three main foundational leading to the most critical experiment foundation for discovery of DNA structure came from X-ray diffraction images, particularly those produced by Rosalind Franklin at King's College London. Key contributions: Franklin's photo 51, 1952. This X-ray diffraction image, taken under high humidity, revealed a clear helical pattern with distinctive X shape, indicating a spiral structure. Spacing of the diffraction spots suggested a repeat distance of 3.4 angstroms between nucleotide bases, a helical diameter of 20 angstroms running out a triple helix, which Watson initially considered two double-stranded double helix structure, as Franklin has deduced before Watson and Crick. Maurice Wilkins. Roll Wilkins, Franklin's colleague, shared her data without her explicit consent with Watson, who recognized its significance. This image was crucial in confirming the helical symmetry and dimensions of DNA. Why it was essential. Franklin's data provided the physical proof needed to reject incorrect models. Example: Linus Pauling's triple helix, and guided Watson's and Crick in constructing an accurate double helix model with correct spacing and orientation of the strands. Controversy and recognition. Franklin. Okay, I do know the woman had a did not have a good life, because yeah, Nobel Prize of Psychology, Physiology of or Medicine was awarded to Watson, Crick, and Wilkins, but not Franklin. Nobel Prizes are not awarded posthumously. Yeah. Now let's read from GPT. Let's start. What is X-ray diffraction? A technique used where X-rays are aimed at a crystallized molecule like DNA. You know, let's check a video if there's any videos. Do Disney Plus.
Glücksgefühl. Für kleine Träumer, für Abenteurer, für euch. Großartige Geschichten. Ein
Großartige
Leben lang.
Geschichten.
Nur auf Disney Plus.
Plus.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time.
I understand.
But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD. And she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny, wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA's structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962
like
for
that,
their
am
work
I
on
right?
DNA. It's often said that
Not
Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice, Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave mother of the double helix.
Rosalind Elise Franklin. Hell yeah. But so I believe that's it. I don't know.
Lebe traumhaft. Auch beim Preis. Lebe smart. IKEA.
Using the right weight matters. The body pod lets you easily understand what is the
Oh.
fastest way to reach your health and fitness goals. At an accuracy of 98%, you can measure 40.
In this video, I will explain Chargaff's rules. In the mid-1900s, Erwin Chargaff discovered a crucial element of DNA structure. Chargaff knew that DNA was made up of 4 different bases: adenine, cytosine, guanine, and thymine. In his experiments, he observed that the ratio of cytosine to guanine was 1:1.
Oh yeah, see that? That's such a good representation of like adenine and thymine goes together, cytosine and guanine goes together. That's good.
He observed that the ratio of cytosine to guanine was 1:1, and the ratio of adenine to thymine was also 1:1. This means that the molecules of DNA he observed were made up of equal percents cytosine and
Someone didn't see what email. Mm-hmm.
and there were also equal percents adenine and thymine. This means that if a DNA molecule has 4 guanine nucleotides, then it must also have 4 cytosine nucleotides. And in the same molecule, if you have 6 adenine nucleotides, then you will also have 6 thymines. This phenomenon can be explained by the complementary base pairing that gives DNA its double-stranded structure. Each strand of DNA is made up of nucleotides, and the 2 strands are connected together by hydrogen bonds linking the bases of complementary nucleotides. Guanine is complementary to cytosine, and adenine is complementary to thymine. So in this double-stranded molecule, everywhere you see guanine, you will find it attached by hydrogen bonds to cytosine. And everywhere you see thymine, you will find it attached to an adenine. That is why Chargaff noted a 1-to-1 guanine to cytosine ratio and a 1-to-1 adenine to thymine ratio in the DNA he studied. Chargaff also discovered that how much of a genome is made up of each of these complementary pairs varies from species to species. One species may have more cytosines and guanines than another species. This discovery led to a better understanding of genetic diversity among different species. For example, the human genome is about 40% guanine and cytosine and about 60% adenine and thymine, while the yeast genome is about 38% guanine and cytosine and about 62% adenine and thymine. So you can see that the amounts of each base vary from species to species. So there you have it. Chargaff's first rule states that in DNA, the number of guanines will match the number of cytosines, and the number of adenines will match the number of thymines. And his second rule states that the composition of DNA varies from species to species. If you'd like to learn more, see the other videos on my channel.
Yeah. A crucial biochemical foundation for DNA structure came from Erwin Chargaff's discovery of key patterns in nitrogenous base composition of DNA. His findings, later termed as Chargaff's rules, provide essential clues about how DNA bases pair. Key observation: analyzed DNA from different organisms and found that adenine equals thymine, guanine equals cytosine, purine equals pyrimidine. Purines are adenine plus guanine. Pyrimidines are thymine plus cytosine. A plus T, G plus C ratio varies by species, but the A equals T and G equals C holds universally. Interesting. Why it mattered for DNA structure: Watson and Crick realized these ratios strongly suggested base pairing. ATGC, yeah, this explained how two DNA strands could be complementary and replicate faithfully. Chargaff's data ruled out models where base paired AAGG, which would not fit the X-ray data. Chargaff's reaction to the double helix: initially skeptical, Chargaff later acknowledged that his rules were key piece of the puzzle. However, he famously disliked Watson and Crick's informal approach, calling them two pitchmen in a search of helix. Okay. Chargaff's work. DNA, the amount of adenine is always equals the amount of thymine. Guanine equals cytosine, suggesting specific base pairing that was essential. What I hate is none of these AI did not talk about the linking between these nitrogenous bases as hydrogen bonds. Both of these did not say about that. Did not talk about that. It's not good. Molecular dimensions. Molecular dimensions. Linus Pauling. Oh, this guy was alive. Linus Pauling influence and model building approach. The third major foundation for the discovery of DNA structure came from Pauling's groundbreaking work on molecular structure and his use of model building as a scientific tool. Alpha helix discovery 1951. Pauling used X-ray data and physical models to deduce the alpha helix structure of proteins, proving that complex biological molecules could be solved through structural chemistry. This inspired Watson and Crick to apply similar methods to DNA. Oh yeah. Oh yeah, it did. It's not boring. It's just a lot to take in. Mm-mm. I believe I should eat something. I do have something to eat. I should eat that because, yeah, I'll be heading to work in about 1 hour. 1 hour, 20 minutes, 15 minutes. Or I would be, I think I'll eat on the last 30 minutes. That's better, I think. But right now, I'm just gonna take a 10-minute break. 10 minutes! This is for real this time. For real, for real. 10 minutes, like I said. Be right back within the 10 minutes, and I am right back within the 10 minutes. So, I've been thinking to change my phone. I'm sorry, I'm going off topic, but I've been using the 7 Plus for a while. Maybe more. I think it's been 2 weeks, 3 weeks. I used the 7 Plus. Why? I don't know. A lot of reasons. I like the design. The the home button. It's a really cool. I should say I really like the home button design. What else? Yeah, mainly it's that. Discuss the design uh, and I want to take a break from my 15 Pro, which is yeah, it's a flagship, but after using maybe for 1 week, you just don't feel anything from this 15 Pro, from almost $1,000, €1,000 device. I don't feel good paying that much money for something. Get some coffee. Drink some coffee and yo. Yo. Well, I decided not to. I'll take today's video. Today I plan to record myself going to work, but I'm gonna record myself on this phone. Let's see if it's good. The rain. Of course it's gonna rain. Download Instagram and TikTok. Oh, come on, down. Fuck with the Wi-Fi. Fuck the Wi-Fi. Oh, that's too bland. That's not too bland. It's okay. It's a drinkable stuff. Both of these should not be here. Oh, I wonder how the TikTok account is doing. I believe it's doing really bad. So, Pauling's discovery was 46 people watched my profile. Okay. Friends don't allow. Yeah. 185 degrees. Okay. The fuck are these? Oh yeah, I do have the TikTok, Instagram. I know I'm wasting time. Just a minute. Just need to check these. So I have decided to make short form content based on something that I study every day. 60 seconds explaining a topic that I learned that day. I think it's a good way to use short form content, right? So if you see if you see this channel, that means after this video you're gonna see more short form content, useful content, not just me talking about my day. So yeah, peace out, people. Yo. Well, I didn't do that. I did not do that. I hate that. I could do it right now, right? I still don't understand what is Pauling's Alpha Helix discovery. That's from Rosalind Franklin, if I believe. Why is it a sexes in proteins prove that helical structure? I still didn't. What is happening to my hair? Why Pauling's DNA model failed? Pauling proposed a triple helix DNA model. Phosphate groups on the inside, impossible due to the negative charge repulsion. Incorrect bond angles. His model was based on dry DNA, not Franklin's hydrated form. Pauling's expertise in hydrogen bonds directly inspired Watson. Key takeaways. So, Let me follow this. Go deep. So these are the base pairs. Sugar phosphate backbone. Phosphate phosphodiester linkage. So hydrogen bond between what nucleotides? I forgot the term. Complementary. Complementary nucleotides and the phosphodiester bonds between phosphodiester bond between bond as strand. Ooh, 2 to 3 kilocalories per mole. Oh, explain why GC is more. Explaining why GC rich DNA is more thermally stable. Link the sugar, deoxyribose, and phosphate groups in the backbone of single DNA strand. Sugar and deoxyribosome. That's the best one. Okay.
Ja, einen schönen guten Morgen. Ich sehe schon, dass die Weltstädte Remscheid und Ida Oberstein hier schon vertreten sind. Köln natürlich auch. Das ist.
It's just beautiful, isn't it? It's mesmerizing. It's double helix sighting. You really can tell just by looking at it how sort of important and amazing it is. It's pretty much the most complicated molecule that exists, and potentially the most important one. It's so complex that we didn't even know for sure what it looked like until about 60 years ago. And so multifariously awesome that if you took all of it from just one of our cells and untangled it, it would be taller than me. Now consider that there are probably 50 trillion cells in my body right now. Laid end to end, the DNA in those cells would stretch to the sun not once, but 600 times. Mind blown yet? Hey, you want to make one?
Make one. Let's look into the phosphodNA.
Of course, you know, I'm talking about deoxyribonucleic acid, known to its friends as DNA. DNA is what stores our genetic instructions, the information that programs all of our cells' activities. It's a 6 billion letter code that provides the assembly instructions for everything that you are. And it does the same thing for pretty much every other living thing. I'm gonna go out on a limb here and assume that you are a human, in which case every body cell that you have, or somatic cell, in you has 46 chromosomes, each containing one big DNA molecule. These chromosomes are packed together tightly with proteins in the nucleus of the cell. DNA is nucleic acid, and so is its cousin, which we'll also be talking about, ribonucleic acid, or RNA. Now, if you can make your mind do this, remember all the way back to episode 3, where we talked about all the important biological molecules, carbohydrates, lipids, and proteins. That ring a bell? Well, nucleic acids are the fourth major group of biological molecules, and for my money, they have the most complicated job of all. Structurally, they're polymers, which means that each one is made up of many small repeating molecular units. In DNA, these small units are called nucleotides. Link them together and you have yourself a polynucleotide. Now, before we actually put these tiny parts together to build a DNA molecule like some microscopic piece of IKEA furniture, let's first take a look at what makes up each nucleotide. We're gonna need 3 things. 1, a 5-carbon sugar molecule, 2, a phosphate group, and 3, one of 4 nitrogen bases. DNA gets the first part of its name from our first ingredient, the sugar molecule, which is called deoxyribose. But all the really significant stuff, the genetic coding that makes you you, is found among the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It's important to note that in living organisms, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. They're like an intertwined microscopic double spiral staircase, basically just a ladder, but twisted, the famous double helix. And like any good structure, we have to have a main support. In DNA, the sugars and phosphates bond together to form twin backbones. These sugar-phosphate bonds run down each side of the helix, but chemically in opposite directions. In other words, if you look at each of the sugar-phosphate backbones, you'll see that one appears to be upside down in relation to the other. One strand begins at the top with the first phosphate connected to the sugar molecule's 5th carbon, and then ending where the next phosphate would go with a free end at the sugar's 3rd carbon. This creates a pattern called 5' and 3'. I've always thought that the deoxyribose with an arrow, with the oxygen as a point, it always points from 3' to 5'. Now on the other strand, it's exactly the opposite. It begins up top with a free end at the sugar's 3rd carbon, and then the phosphates connect to the sugar's 5th carbons all the way down. And it ends at the bottom with a phosphate, and you've probably figured this out already, but this is called the 3' to 5' direction. Now it is time to make ourselves one of these famous double helices. These 2 long chains are linked together by the nitrogenous bases via relatively weak hydrogen bonds. But they can't be just any pair of nitrogenous bases. Thankfully, when it comes to figuring out what part goes where, all you have to do is remember that if one nucleotide has an adenine base, only thymine can be its counterpart. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. The GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases, or the base sequence, that allows your DNA to create you. So AGGTCCATG means something completely different as a base sequence than, say, TTCAGTCG. Human chromosome 1, the largest of all of our chromosomes, contains a single molecule of DNA with 247 million base pairs. If you printed all of the letters of chromosome 1 into a book, it would be about 200,000 pages long. And each of your somatic cells has 46 DNA molecules tightly packed into its nucleus. That's 1 for each of your chromosomes. Put all 46 molecules together, and we're talking about roughly 6 billion base pairs in every cell. This is the longest book that I've ever read. It's about 1,000 pages long. If we were to fill it with our DNA sequence, we'd need about 10,000 of them to fit our entire genome. Pop quiz. Let's test your skills using a very short strand of DNA. I'll give you one base sequence. You give me the base sequence that appears on the other strand. Okay, here goes. So we got a 5' AGGTCCG to 3'.
Oh shit.
And time's up. The answer is 3' TCCAGGC.
Fuck me. So A goes to ATGC, yeah. Five to three, 3. Go from here. It's 3 to 5. I know this one. It's TCCA. Oh, TCCAGGC.
Times up, the answer is 3 prime TC
Yeah.
AGGC 5 prime. See how that works? It's not super complicated. Since each nitrogenous base only has one counterpart, you can use one base sequence to predict what its matching sequence is going to look like. So, could I make the same base sequence with a strand of that other nucleic acid, RNA? No, you could not. RNA is certainly similar to its cousin DNA. It has a sugar phosphate backbone with nucleotide bases attached to it, but there are 3 major differences. 1, RNA is a single-stranded molecule, no double helix here. 2, the sugar in RNA is ribose, which has one more oxygen atom than deoxyribose, hence the whole starting with an R instead of a D thing. And finally, RNA does not contain thymine. Its 4th nucleotide is the base uracil, so it bonds with adenine instead. RNA is super important to the production of our proteins, and you'll see later that it has a crucial role in the replication of DNA.
Hier sind 3 Gründe, warum dein Vertriebsteam das Monday Sales CRM gerne nutzen wird. Erstens, es ist total einfach.
DNA. But first, biographies. Yes, bandages from a nearby hospital. God. First, sure was not possible, and in doing so, she indicated that DNA may indeed
I do.
copies itself because cells are her contribution. Even worse than that, the Nobel Prize Committee couldn't even consider her for the prize that they awarded in 1962 because of how dead she was. The really tragic thing is that it's totally possible that her scientific work may have led to her early death of ovarian cancer at the age of 37. At the time, the X-ray diffraction technology that she was using to photograph DNA required dangerous amounts of radiation exposure, and Franklin rarely took precautions to protect herself. Nobel Prizes cannot be awarded posthumously. Many believe that she would have shared Watson and Crick's medal if she'd been alive to receive it. Now that we know the basics of DNA structure, we need to understand how it copies itself, because cells are constantly dividing, and that requires a complete copy of all that DNA information. It turns out that our cells are extremely good at this. Our cells can create the equivalent of 10,000 copies of this book in just a few hours. That, my friends, is called replication. Every cell in your body has a copy of the same DNA. It started from an original copy and will copy itself trillions of times over the course of a lifetime, each time using half of the original DNA strand as a template to build a new molecule. Helicase is marvelous, unwinding the double helix at breakneck speed, slicing open those loose hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication fork. It has a top strand called the leading strand, or the good guy strand as I call it, another bottom strand called the lagging strand. These unwound sections can now be used as templates to create 2 complementary DNA strands, but remember, the 2 strands go
subtitles,
in
the
opposite
captions
directions
are
in terms
so
of their chemical structure,
laggy.
which means that making a new DNA strand for the leading strand is going to be much, much easier than for the lagging strand. For the leading, good guy strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down the molecule. But before it could do that, it needs a selection of nucleotides that fill in the section that's just been unzipped. To get started at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer, just a little thing for it to hook onto so that it can start building the new DNA chain. And for that little primer, we can thank the enzyme RNA primase. The leading strand only needs this RNA primer once at the very beginning, then DNA polymerase is all, I got this, and it just follows the unzipping, adding new nucleotides to the chain continuously all the way down the molecule. DNA polymerase can only copy strands in the 5', 3' direction, and the lagging strand is 3', 5'. So DNA polymerase can only add new nucleotides to the free 3' end of a primer. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments. And here, that awesome little enzyme, RNA primase, does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, each starting with an RNA primer. These are called Okazaki fragments, after the couple of married scientists who discovered this step in the process in the 1960s. And thank goodness they were married so that we could just call them Okazaki fragments instead of Okazaki-someone's-someone fragments. These allow the strands to be synthesized in short bursts, and then another kind of DNA polymerase has to go back over and replace all of those RNA primers, and then a little fragments gets joined up by a final enzyme called DNA ligase. DNA replication gets it wrong about 1 in every 10 billion nucleotides, but don't think your body doesn't have an app for that.
The subtitles, fuck that.
But the DNA polymerases can also proofread, in a sense, removing nucleotides from the end of a strand whenever they discover a mismatched base. Because the last thing we want is an A when it would have been a G. Considering how tightly packed DNA is into each one of our cells, it's honestly amazing that more mistakes don't happen. Remember, we're talking about millions of miles worth of this stuff inside us, and this, my friends, is why scientists are not exaggerating when they call DNA the most celebrated molecule of all time. So you might as well look this episode over a couple of times and appreciate it for yourself. And in the meantime, gear up for next week when we're going to talk about how those 6 feet of kick-ass actually make you you. Thank you to all the people here at Crash Course who helped make this episode awesome. You can
My work is easy.
Bro, just get the fuck out of here. There's no no playlist? There should be some fucking playlist up here.
hello and welcome to
Where's the playlist? Well, people, I'ma go to eat my food. It's food time. It's Essenzeit. Es ist Mahlzeit, da. Mahlzeit, ja. Yeah, I think it's better to end the stream. I couldn't do too much today. I hate myself for that. Whatever. Ozaki fragments, structure of chromatin. Yo, I gotta eat.